Chapter 22, The Immune System, Part 1, Screencast 3. This last screencast here will focus on nonspecific internal defenses, mostly two major physiological defenses that we have, and those are the inflammatory response and also fever. Now, inflammation is an immediate response to ward off unwanted substances. Local, nonspecific response of vascularized tissue to injury. So keep in mind, the inflammatory response is limited to well-vascularized tissue. That is a really important part of this. And remember, this is a part of innate immunity, so it will respond to a number of different types of attacks upon the body. Now, when we look at the events of inflammation, the first part here is going to be some type of injury to the tissue. We're going to have various types of cells involved in response to this, such as basophils, mast cells, and infectious organisms, and they are going to be releasing chemicals that are responsible for the initiation of this response. Chemicals that play this role include histamine, leukotrienes, prostaglandins, and chemotactic factors. We will describe some of these terms as we move on to this screencast. Now, there are a number of major events that are part of the inflammatory response, so we're going to introduce these events and then we're going to put them together on a diagram that goes over this in great detail. So the first part, there are chemicals that are released which are going to cause vascular changes. These changes will include, but are not limited to, vasodilation, increased capillary permeability. So this means that it will be easier for things to enter and leave the capillaries. And also there is going to be increased endothelial expression of molecules that are necessary for the adhesion to white blood cells. These are known as cell adhesion molecules, or CAMs. The next part will be the recruitment of leukocytes, and this is divided into three separate processes. The first is margination, and this is when white blood cells will actually adhere to these endothelial adhesion molecules, and they're basically going to be rolled along the wall of the capillary. Diapedesis, this is the actual process in which how cells will escape blood vessel walls. And then we have chemotaxis. Leukocytes will migrate towards these chemicals that are released from damaged, dead, or pathogenic cells. So think of these chemicals as white blood cell magnets, and they're going to draw white blood cells towards the site of injury. So examples here would be white blood cells that are releasing cytokines, and this is going to stimulate the process of producing more white blood cells in red bone marrow. Macrophages, they may actually release pyrogens, and these are going to be molecules that specifically result in a fever. There's going to be the delivery of plasma proteins to the site of injury. So examples of this would be immunoglobulins, complement, clotting proteins, and kinins. Clotting proteins form clots that will wall off microbes. Kinins actually stimulate pain receptors. They'll have an impact on changing and by increasing capillary permeability. They also will increase the production of cellular adhesion molecules, or CAMs, by capillary cells. Now, kinins are produced from inactive kinagens, and these are originating from the liver and other cells. An example here would be bradykinin. Now, the last we actually have is this fluid, or this exudate, which we'll refer to it, and that actually moves from blood to injured or infected area. This is going to be fluid, protein, immune cells to eliminate the pathogens and promote the healing process. We'll have the process of vasodilation, which is going to bring more blood to the area of infection. The contraction of vessel endothelial cells open gaps between them, and here's a further increase in capillary permeability. We will see the loss of plasma proteins, so this is going to decrease capillary osmotic pressure, so it's going to decrease fluid reabsorption into the blood. Now, the extra fluid is taken away by lymphatic capillaries in the area. So this is, think of it as a washing or cleaning up at the site of injury. So what's happening here is actually the carrying away of debris, and it allows lymph nodes to monitor its contents. generally there'll be a decrease within 72 hours of the inflammatory response. During this time, we can see macrophages eating bacteria, damaged host cells, and dying neutrophils. And also, tissue repair begins as fibroblasts and the formation of new connective tissue at the site of injury. Now here we can actually see all of these processes taking place. So part one, remember, was the release of inflammatory and chemotactic factors. So we have mast cells, and they're going to be releasing primarily histamine. Okay, we are going to see changes in the vessels, and this includes vasodilation of arterioles, a change in capillary permeability, so capillaries will become more leaky, and also we're going to see the display of these cellular adhesion molecules. Then we have the recruitment of immune cells, and we can actually see what's happened to the white blood cells. Notice they have adhered themselves to the cams, and they're just going to roll along the vessel. And how they're actually going to leave the vessel is through the process of diapedesis. And we can see that here as they're squeezing through these intracellular gaps. Now, the last process is chemotaxis. And remember, what we have going on here is a number of different types of chemical agents that are released from the site of injury, and they're actually going to be drawing towards them white blood cells and other immune cells overall. Then the last process is that delivery of plasma proteins. So notice we have the proteins which will be leaving. This is going to decrease osmotic pressure. So this is going to be one of the reasons why we quite often will see swelling at the site of injury. And the last role or part of the inflammatory response is the formation of exudate. And remember, this is the washing away of the infected area, the cleanup process, so to speak. And all of this is going to be accomplished by lymphatic capillaries. So this basically, this excess fluid will be taken up by lymphatic capillaries, and it will be monitored within the lymphatic system. Now, when we look at the inflammatory response or inflammation, there are 4 general signs that we refer to as the cardinal signs of inflammation. They are redness, and that is due to the increased blood flow. There's heat, that is due to the increased blood flow. And also remember that this is all examples of metabolic activity and will be generating heat also. There will be swelling, and remember there's going to be excess filtration because there's going to be a loss of proteins within the capillaries. So we'll have that accumulation of fluid at the site of injury. And then last but not least, we'll actually have pain, and this is due to the stimulation of pain receptors. So remember, we've got this extra fluid and these chemical irritants. They could be activating pain receptors and having those impacts overall. Now, keep in mind, loss of function is not considered to be a cardinal sign. It is actually a result of pain and swelling in severe cases. A really good example is loss of range of motion when someone sprains their ankle. Now, when we look at how long the inflammatory response takes place, it generally lasts anywhere from 8 to 10 days. Now, we do have something known as chronic inflammation, and this is an inflammatory response that goes on for an extended period of time. And we know that chronic inflammation has significant detrimental effects on the body. Now, the last nonspecific internal defense mechanism that we are going to address is the fever. Fever, known as pyrexia, is an abnormal increase in body temperature. Typically, we consider a fever to be 1 degree Celsius or more from normal, and remember, normal is 37 degrees centigrade. Now, this is actually caused from the release of substances called pyrogens. So these are a type of interleukin, and they are coming from immune cells or infectious cells. Now, when we look at the events that take place during a fever, pyrogens actually circulate through the blood, and the target is the hypothalamus. We've talked quite a bit about the hypothalamus in this course, and we need to remember that there are a lot of behavioral and control mechanisms that are housed within the hypothalamus, and this is actually, we want to think of as the body's thermostat. So in response to these pyrogens, the hypothalamus will release prostaglandins. And this results the hypothalamus to raise the temperature set point, and this allows body temperature to increase. There are 3 stages of a fever, and they are known as the onset, stadium, and defervescence stages. Onset is when the body temperature actually begins to rise. And what the hypothalamus is doing in this situation, it is stimulating the constriction of blood vessels within the skin. Remember, this is a major mechanism in how we release heat to the external environment and maintain normal body temperature. So now we're not going to be able to do this because these blood vessels are constricted. It also will trigger the shivering of muscle. And remember, the generation of heat as a waste product of muscular contraction, and this could be in response to chills, which we know is something that accompanies fever. Stadium, this is the stage in which the elevated temperature is maintained for a period of time. During this time, metabolic rate increases, and this is to promote elimination of harmful substances. We also will see that the liver and spleen will bind zinc and iron, and this actually will slow microbial reproduction. The final stage is defervescence. This is when the temperature returns back to normal. The hypothalamus is no longer producing or being stimulated by pyrogens. There's going to be a decrease in the release of prostaglandins, and then the hypothalamus will stimulate mechanisms to release heat. So now, instead of vasoconstricting, they're going to vasodilate blood vessels of the skin and actually promote sweating. Now, when we look at fever, it's important to realize that there are going to be benefits and consequences. So one of the things that's important is to leave a low-grade fever untreated, because there are a number of benefits associated with this. Some of them include, but are not limited to, the inhibition of the reproduction of bacteria and viruses. Now, keep in mind, this elevated temperature is not sufficient to kill bacteria and viruses, but it actually will decrease the rate at which they're going to be reproduced. We'll see an increase in the activity of interferon. We're going to see an increase in the activity of adaptive immunity in such a way that it is supporting what is happening with respect to the fever or what is causing the fever. There also will be an acceleration of tissue repair. The other thing that might happen is an increase in the cellular adhesion molecules on the endothelium of capillaries within lymph nodes. Now, what this means is additional immune cells which will be migrating out of the blood. As I said earlier, when we look at a low-grade fever, it is best to leave it untreated. Now, there are major risks associated with a high fever. High fevers can be potentially dangerous, 103 degrees Fahrenheit in children, and probably a little bit lower than that in adults. Now, what's happening here is changes in metabolic pathways, and remember, proteins in particular are sensitive to increases of heat, and if we have a high fever, we may actually promote the denaturation of proteins. And remember, when you change the shape of a protein, it's no longer going to work effectively. Additional consequences include seizures. Now, if we look at a fever that is greater than 106 degrees Fahrenheit, the result will be irreversible brain damage. And if we look at a fever greater than 109 degrees Fahrenheit, death is especially likely. Now, we kind of talked about exudate before, but now we actually have pus. And this, remember, contains destroyed pathogens, dead white blood cells, macrophages, and cellular debris. Typically, this should be removed completely by the lymphatic system or through the skin. Now, if that does not happen, the potential for an abscess or infection to take place. So now the pus is actually walled off by collagen fibers, and this generally may actually require surgical intervention to remove the abscess. Now, one of the things that if you have taken a first responder first aid course is introduced to ice. Ice is something that is recommended for acute inflammation, and what it does, it actually causes vasoconstriction of blood vessels. So it turn, you're actually decreasing the inflammatory response. But what really is happening here is you're numbing the area, and that actually makes it less painful. Now, we introduced the concept of chronic inflammation previously, and chronic inflammation is considered to be an inflammation event that lasts for more than 2 weeks. It's going to be characterized by macrophages and lymphocytes, no, not neutrophils. Now, I'm going to use the example of overuse injuries as a different type of chronic inflammation. This is usually due to some issue of biomechanics or overuse or something like that. Examples could be shin splints, tennis elbows, those types of things. What I want to address more often, which is a greater concern, are these inflammatory responses that are actually associated with autoimmune disorders. And the issue here is they can actually lead to the destruction of tissue, formation of scar tissue, okay? And also another thing that might stimulate this chronic inflammation is the inflammatory response is just not capable of eliminating the pathogen in question.